Hi everyone. Now we are going to discuss about the SAR of antihypertensive agents. Under this, we are going to discuss about this SAR of ACE inhibitors. Normally, before going to that topic, we have to know what is hypertension. That is, systolic and diastolic pressure exceeds 140/90 mm/mercury. That is known as hypertension. And the drugs which are used for the treatment of hypertension are called as antihypertensive agents. So, ACE, what do you mean by the term ACE inhibitor? That is angiotensin converting enzyme. That will be inhibited. Angiotensin converting enzyme ACE is a zinc containing glycoprotein. So this it is a glycoprotein in protein in nature. That is an example for glycoprotein. That is also containing a zinc metal. The important binding site of ACE are cationic site to attract the carboxylate ion. So normally the important site of this one is it is a cationic site that contains it is used to attract the carboxylate ion. Zinc ion that can polarize the carboxyl group of amide function to make it more susceptible to hydrolysis. The main function of this zinc ion is it can polarize the carboxyl group of the amide that is used to make it more susceptible for the hydrolysis. So this is the basic nucleus, that is benzene, that is phenyl group CH2N, then zinc binding group X usually it will be a methyl group, C double bond O, then N3 will be present in that one. This zinc binding group it should contain a sulfonyl group, then carboxylate, that CHCOO, it's like the carboxylate group, then phosphate group. If it is present, then the compound will be binding to that zinc containing group. So among the three zinc binding group, sulfur hydroxyl group that is superior. So we have discussed about these three groups, that is sulfonyl group, carboxylate group, phosphate group. Among these, this sulfur hydroxyl group is superior one, but they form disulfide, which may result in shorter duration of action. We have discussed about the three groups, but sulfur hydroxyl is the superior one, but it is having very short duration of action. So N ring must contain a carboxylic acid group. If you see that N ring, it should contain. We will see the structure at that time. We can understand that N ring should contain a carboxylic acid group to mimic the C-terminal carboxylate of the ACE substrate. So if it is containing a carboxylic acid group like this, it will mimic the action of that ACE, that is angiotensin converting enzyme substrate. Then the larger hydrophilic heterocyclic in the N ring increase the potency and alter the pharmacokinetic properties. So if you are substituting with a heterocyclic atom, that is hydrophilic in nature, that will increase the potency and it is also increase the pharmacokinetic properties. Then this X is usually a methyl group which is mimics the side chain alanine of ACE substrate. See, normally here we have already discussed about this one. The X should be a methyl group, usually a methyl group that will mimic the side chain alanine of the ACE substrate. Then when the stereochemistry of inhibitor is consistent, that is either it may be a levorotatory or dextrorotatory. If it is the stereochemistry is consistent, then optimum activity occurs with L amino acid. If it is a L form of that amino acid, then the activity will be more. This is one example, that is enalapril. It contains C6H5, CH2, CH, COO, C2H5, NH, CH, CH3, CO, then pyrrolidine, 2 carboxylic acid will be present in that one. This is one example. You can find all these structures. That is amino group. Okay, then heterocyclic atom with the carboxylic acid group. This is the example for this SAR activity. Then captopril it is also one of the example, that is it is containing a pyrimidine nucleus. In second position carboxylic acid group is present. Then sulfhydryl group is, that is sulfonyl group is present. CH2SH is present. So this is one of the example. All these substituents will be present. That is sulfonyl group, carboxylic acid group. Then that if it is substituted with some other phosphate group, that will give the better activity. This is the SAR of ACE inhibitor.